Okay, we can start the next session. Now we have the short talk, and there are posters that have been selected for presentation. And the first one is Alaine Marsden from Johannesburg. So please, GP, you have to take down the presentation, the slide.
Okay.
Thank you very much.
Hello there, my name is Alaine Marsden, and I'm a master's student being supervised by Dr. Catherine Skeepers at the NICD in Johannesburg. And today I'm going to be presenting the subject of my master's dissertation, which is diversity found in the IGH locus of a South African subpopulation. So the human IGH locus, or immunoglobulin heavy chain locus, is found at the very tip of chromosome 14 and contains hundreds of genes that code for the heavy chain of an antibody, which plays a critical role in antigen recognition and effector function. So it follows that polymorphisms found in this area will have an effect on an adaptive immune response. And many of these genes have multiple alleles, leading it to be one of the most polymorphic complex loci in the human genome. So we have previously done some work in a South African population, looking at IGHV genes in 28 women from rural and urban KwaZulu-Natal. These women are mostly Southern Bantu Zulu-speaking women. And what was really striking in that study was that 48% of the alleles had never been described before. So we're looking to expand on this work. And when the study was conducted, we were really only looking at the V region, that blue X on there, and the figure, which is responsible for antigen recognition. So we wanted to now look at both the leader sequence in yellow, which modulates antibody expression and secretion, and the black RSS, or recombination signal sequence, which affects how antibody genes are stitched together to make a final antibody. So the first aim of the work was to investigate IGHV polymorphism in a larger group and across the whole gene. To do this, we performed an amplicon sequencing approach on MySeq and PacBio, looking at 70 participants from the same population. And this graph shows you all the genes that we're able to amplify and sequence in the study. And so the blue, the black alleles are those alleles that matched IMGT alleles. Those blue alleles are those that matched sequences from IGPDB, which contains truncated genomic DNA or data from inferred allele studies. So we're able to actually now provide full-length IGHV sequences for those. And then really interestingly, in yellow are novel sequences. And this accounted for 20.5% of the alleles that we saw. And these were novel in addition to the previous novel diversity we had seen in those 28 participants. And that was really interesting. And the next thing that we wanted to look at within our kind of NGS approach was to look at variation in the regulatory regions, which we defined in two ways. The first was single nucleotide variation, kind of depicted on the left-hand side of your screen, where participant leader sequences may have had little nucleotide differences from the reference. And then on the right-hand side, there was a mismatch because upon further investigation, some of these sequences actually just matched other alleles. So what you're looking at here is IGHV 1-18, where most of the sequence in our participants actually matched the 0-1 allele, but the leader matched the 0-4 allele. And when we looked at the prevalence of this diversity, we were able to see that it was actually surprisingly common, with 44.8% of unique sequences displaying some kind of variation, particularly in the leader sequence or the intron. And this graph on the right-hand side shows you the distribution. It's a bubble plot where the size of the bubble for each different kind of variation indicates the number of individuals it was found in, and how high that bubble is the percentage of unique sequences that was found in. And overall mismatch was surprisingly common, especially in the intron. And so we hypothesized that this variation might have some kind of effect on how antibodies are secreted and expressed. So our next aim was then to take a step back and look at the entire locus, and really to look at copy number variation and structural diversity. So the IGH locus is interesting in that it exhibits a lot of structural variation, which causes differences in copy numbers of different IG genes. And what you're looking at here is how a person, even in their different haplotypes, may have different insertions and deletions when you compare to a reference chromosome. And the first thing we did was to take our NGS data and count the number of unique alleles found for each gene in a particular person. And we just wanted to, first off, look at commonly duplicated genes. And we saw that there was duplication that was common for us in IGHV 169 and IGHV 2-70, especially the former, which was duplicated in more than 50% of our participants. But then when we took a further look, we found that there were genes that were duplicated that haven't yet been reported in literature, although at relatively low levels. So we wonder if this is maybe a population signature or indicative of more southern Bantu samples. But a targeted NGS approach is not sufficient to really capture the entirety of structural diversity. So I was very fortunate to travel to the University of Louisville, spend some time in Dr. Corey Watson's lab, where they have pioneered an IGH hybridization capture technique. He's actually presenting a poster on this at this conference if you're interested. And it essentially uses magnetic probes to isolate IGH fragments from shear genomic DNA, and these fragments are quite large. They can be up to 8 or 9 kb, which were then sequenced on a PacBio and reconstructed computationally to get a look at the locus. So we had DNA from 2 of our participants that we performed this technique on, CAP88 and CAP255. And just by comparing them to a reference, we saw that there were a lot of single nucleotide variants across the entire locus. Most of these lay in intergenic regions, in the spaces between genes. However, there were several that laid in coding regions and regulatory regions, kind of emphasizing and corroborating what we had seen before in the NGS data. We were also able then to look at also indels of varying sizes, and these were also present across the loci. And some of these were incredibly large, encompassing multiple genes. And with all of this data, we were able to construct a schematic of the loci in these individuals. So what you're looking at here is CAP88, CAP255, having rows of boxes that represent chromosomes, and each box represents a gene position. The color of the box represents whether they're homozygous, heterozygous, or hemizygous at the position, or whether there was the presence of a novel allele. And not only are there differences between the haplotypes of these individuals, there are also really, really large structural differences, which are indicated here in green and in blue, I mean green and red. And particularly on the right-hand side, there are these stretches of 3-4 genes that are inserted in CAP88, but actually aren't inserted at all in CAP255. There are gaps currently, across the locus, and that's because this is a fairly early run of the technique. But even with patchy data, we were able to get a really comprehensive look at structural diversity across the loci of these individuals. So in conclusion, we were able to detect even more novel alleles than we saw previously, kind of indicating that we're always at the tip of the iceberg when it comes to African genomics. And we were able to provide full-length data for several IGPDB alleles. And we were also able to look at variation in lead sequence in a lot of individuals and found that there was frequent mismatch between the leaders of different alleles. We were also then able to look at copy number variation and structural diversity and see that not only was duplication really common, but also there were these broad structural differences between just 2 individuals from this rather complicated population. So the phenotypic implications of this diversity are kind of the next logical question, and we're already beginning to work on that within certain disease contexts like HIV to examine how polymorphisms may create differential immune responses. So thank you to all of our funders and to everyone that made this work possible.
Thank you, Alaine. Nice talk. We have a couple of questions. One from an anonymous. Did you perform a repertoire sequencing of the 70 participants, or did you target amplification of the V gene only?
So the sequencing was germline sequencing, targeting IGHV genes from germline DNA.
Okay, and we have another question from Colin. Have you looked into genetic variation of the SC region?
So we do have some projects currently ongoing looking at variation on IgG3 genes and IgA genes, and whether those have any impact on FC function. So there are projects currently ongoing looking at that.
And I have a question. The two donors that you sequence with PacBio, they are from the same cohort that you sequence in South Africa? Yes. And I don't know if I saw correctly, but I saw that one of the two have both 434 and 439 at least deleted. Is that correct?
So they weren't deletions, but they're actually just missing data. Drops.
Okay, they're missing data. Yeah, because that was surprising at least for me. But okay, thank you very much. Okay, we can move to the next speaker.